Thank you, Sarah. Please join me in thanking Sarah.
The last speaker for this session will be Clemens Falker-Gieske from the University of Göttingen, and Clemens will be talking about the discovery of a genetic variant in serum amyloid A gene that might protect chickens from amyloid arthropathy. Clemens, the floor is yours.
Thank you. Thank you for giving me the opportunity to talk here at this conference. So today I want to convince you that we discovered a mutation in the serum amyloid A gene that at least has a modulating effect on a disease in chickens which is called amyloid arthropathy. So AA is characterized by swollen knee joints in the affected birds, as you can see here on the upper picture on the right. This leads to lameness in these birds and eventually to some of them to death because they can't reach their food. Inside the knee joints, you find these protein aggregates made up of serum amyloid A protein, as you can see in the lower left picture. And the interesting thing about this disease is that brown egg-laying chickens are predominantly affected, while white egg-laying chickens are obviously resistant to the disease. So it is known that these two chicken lines have a differential immune response to bacterial infection, which is actually the cause of the disease. So in brown layers, you see a humoral antibody response, and in white layers, it's a cell-mediated immune response. But the genetic component that makes the difference between the two chicken lines in regard to disease susceptibility is still unknown. So first, some words about the protein that is causing this disease phenotype. So serum amyloid A is an acute phase protein, meaning it's highly upregulated during an insult to the organism, which could be infection or an injury, for example. It's primarily produced in the liver, and in the liver it associates to so-called high-density lipoproteins. So that's the structure you see on the right, this little ball there. Once it is incorporated in such an HDL, the protein travels to the site of infection and then has some downstream effects in inflammation and in the immune response. So we were interested, of course, in the genetic difference between the two lines that makes the white layers resistant. So we received 50 whole genome sequences from the group of Professor Semjana, with which we could compare 25 brown layers to 25 white layer genomes. And first we looked on the chromosome where the SAA gene is located, which is the chromosome 5. So we found an average fixation index of 0.355. And inside the coding region of the SAA gene, we found numerous variants that had an FST above 0.8. And one mutation especially caught our attention, which is on the upper left, this RS739 and so on. So this is a missense variant in the 4th exon of the gene. And most of the white layers carried the allele, and we couldn't find it in the brown layers. So what does this variant actually do? So it is predicted that this leads to an arginine to serine exchange at position 90 in the protein. And on a biochemical level, that means that this protein loses a positive charge and gets more hydrophilic. So this is important later on in the talk. Even though it had a pretty low SIFT score, so it was predicted not to be very severe for the organism, we decided to do some experiments with this variant. So what I did, I created 7 different cell lines which stably overexpress different vector plasmids. So one cell line contains empty vector, 3 cell lines contain the wild-type isoform of the SAA, so the brown layer isoform, and 3 contain the mutant, so the white layer isoform. As you can see in this graph, they more or less all have the same expression level, even though we saw a slightly higher expression of the wild type on the RNA level. So this is then completely the opposite on the protein level. So we ran some Western blots on these cell lines, and as you can see in the upper left plot, we get a lot more protein expressed in the mutant cell lines with white layer isoform compared to the wild type. Looking at the extracellular space, we find only SAA protein in the case of the mutant isoform. So there was no wild type SAA extracellularly with the brown layer isoform. So this was a very interesting finding. So we thought we'd dig a bit deeper. And as you've seen in the beginning, in the knee joints, you see this aggregated SAA. So we were interested in the aggregation property of the two isoforms. So we took cell lysates again and treated them with a biochemical protocol with which you can separate your cell lysates into a detergent soluble and a detergent insoluble fraction. And in the insoluble fraction, you would expect to find aggregated protein. So to our surprise, we only found insoluble protein in the white layer isoform. But if you remember from the beginning, the protein aggregates in the knee joints of brown layers. So we are a bit puzzled about this result. We did some further aggregation assays, so we tested different protocols, and it turns out that the stuff that is found in the white layer version of the protein is not amyloid. So we think it takes up a better sheet-rich conformation, but does not aggregate to a larger amyloid entity in those cells, at least. To further characterize the cells, we did some immunofluorescence stainings to see where the SAA is located and how it looks like. And also, what was very striking, you can especially see it in the lower cell line here, which expresses the white layer version, that we see this kind of spotty localization of the SAA. So if a protein gets insoluble, if you look in the literature, it means it will be in inclusion bodies. Inclusion bodies is a biological term which is very loose, or it doesn't really describe a cell compartment or something. But it has been published in 2017 that SAA aggregates are degraded in the lysosome, and we're currently investigating if these spots that we see there are actually lysosomes. So another strategy that we followed then was to compare these cell lines with RNA-seq. And this was also a very striking result. So as you can see on the left, we have a lot more differential expression with the wild-type isoform of the protein compared to the mutant form on the right. And it was even more surprising that we only have 74 transcripts in common differentially expressed between the two cell lines. So again, this was a very puzzling result. And we dug a bit deeper. So by running protein-protein interaction analysis, I found a huge cluster of transcription factors. This is these here, what you see on the left side. And the interesting thing is that most of these transcription factors are downregulated in the wild-type version of the protein. We're now expressing that. But it's not significant in the mutant version. Some are, but a few lost statistical significance along the way. These transcription factors are mainly have the function of modulating cell fate determination. So SAA-R90S partially lost that effect on transcription factor expression, and it gained a lot of functions. As you can see with the geoterm analysis, the function of this protein rather shifted to the cell membrane. So we found a lot of genes differentially expressed that are located at the cell membrane, the cell periphery, and the extracellular space. And we found none of these genes in the wild type, not even a single one. So it's kind of proves what we've seen in the Western blots, that the protein goes out of the cell. So we thought about what that means for the disease. So it is known in mice that if you add the cytokine IL-6 together with SAA on naive CD4 T immune cells, they differentiate into pathogenic Th17 cells, which also plays a role in rheumatic diseases, for example, which are pretty similar to that disease in the chickens. So I remapped the results from that cell study with our pipeline and found an overlap of 24 differentially expressed genes with the SAA wild-type cells compared to this mouse experiment, and only 8 overlapped with the mutant form. And among them were 2 transcription factors, which again are important for cell fate specification and commitment and determination. So we think that in the SAAR90S version, this property of inducing cell fate determination is lost, and this is something we're going to investigate in our next studies. So summarizing, we think that we found a mutation that at least modulates the response of chickens to the bacterial infection, and in turn modulates the amyloid arthropathy. Or it could even make chickens resistant. We have to check if probably other alleles are involved. On a functional side of the whole story, we think that due to the loss of charge and the loss of lipophilicity, SAA is not integrated into high-density lipoproteins anymore and can't travel to the site of infection and hence cannot aggregate in the knees. And the last point that I already mentioned is that we think that this mutant SAA isoform lost the ability to induce a cell fate change in T cells, and so the immune response between the 2 lines differs because of this. So I'm open for questions now, and thank you for your attention.
Thank you, Clemens. Thank you, Clemens, for an interesting talk, which is now open for questions. While we are waiting for questions, I may start. So I was wondering why you focused on the missense mutation and not on the splice site mutation, which also showed a very strong differentiation.
Well, that's just what we focused on first. So all those mutations are still on the list of things we want to look at. And you're right, this splice acceptor mutation is also very interesting.
We have a question, I believe, Barbara.
So finally, it's a loss of function, so it's recessive?
We don't know. So I don't know if a heterozygous condition is sufficient. Probably partially. I don't know. We haven't tested that yet.
We have time for 1 or 2 more questions.
Thank you. The SSA protein is very important in the acute immune response. So is there any adverse effect of loss of function?
I would assume so. So the question is here that we are asking us, so if that mutation is protective against that disease, does it alter the immune system of the chickens in a negative way? So that's, of course, a point we have to address. So if you think about it, if SAA does not travel to a site of infection, those animals have a completely different immune response to infection. And that's what people saw in the experiments, that the brown layers have a humoral response. The white layers is cell-mediated. Probably that's the key to the difference. We don't know yet. We will test that. Yeah.
Do we have any other questions? If this is not the case, then please join me in thanking Clemens.
Thank you.